Hello everyone, I'm Gil-Je from Akoya Bioscience. Thank you so much for your invitation to FIMSA and give a chance to introduce to Codex technology from Akoya Bioscience. Today, I will talk about the importance of the spatial biology to understand complex immune network and find important clinical clue using Codex. My talk will start with what is spatial biology and how it is different from other technologies to understand the immune network in complex tumor microenvironment. You may be experienced in the NGS. The left panel that represents the historical view of bulk tissue analysis like DNA or RNA sequencing. This approach delivers huge transcriptomic and genomic information without any spatial correlation within cells. Recently, some technologies represent ROI cell analysis using some images. You can see the concept in the middle. It brings puzzle pieces with images, but it still means key pieces. It means the connections between cells within single cell level. True single cell spatial biology with high resolution images allow you to see cellular relationship. I mean different functions in different cellular neighborhood. You can see the clear concept of the spatial biology in the right panel. The context from the images describe how cells interact and have a relationship to each other. So the spatial biology with the single cell analysis and the high resolution imaging, it can give much more and accurate information about complex immune response than other existing technology. So I can say that Akoya Bioscience is a leading company in this spatial biology world. I will introduce Codex system, the best technology for this spatial analysis from Akoya. Codex was designed to address the need for this spatial analysis of single cell on tissue. We have validated the Codex system as a part of the complete solution that include optimized antibody and reagent, then sophisticated automatic fluidic system, and the software suite for downstream imaging processing and analysis. These three pillars are essential for successful and seamless experiment for multiplexity. At first, I will explain the Codex chemistry, how Codex identify over 40 antigen in single cell section. The key factor of the specificity is the unique oligonucleotide barcode conjugated to each antibody. The various nucleotide sequence of barcode give a source to detect specific antibody. To detect each barcode on antibody, Codex uses dye-labeled reporters, which is complementary nucleotide to barcode. Codex uses 3 dyes in single reaction. These dyes are spectrally separated, and it allows clear signal detection without any further unmixing step. This base pairing between barcode and reporters make the key how Codex detect antibody on tissue. Now, you may have questions. How Codex identify over 40 markers using only 3 dyes? It is available from repeated staining cycle with reveal, imaging, and remove of the probe. This is the answer to your question. Please see this animation about repeated Codex staining mechanism. To make this imaging cycle, Codex autostainer need to be integrated with the microscope. As a basic model, Codex uses Keyens microscope. It's enclosed microscope to protect frozen sample from the light, and it has very robust auto-imaging stage. Codex require reagent preparation and manual antibody staining at runtime. This preparation step is done the outside of the Codex, and then the staining the stage will be with the sample is inserted in the Keyens microscope. All staining cycle is performed inside of the microscope automatically. It's controlled by the Codex software. Staining stage is designed to stain cover slip over within two gaskets. The important thing you have to remember is that you have to prepare the tissue sample on cover slip, not slide glass, for Codex learning. This is a reconstructed move with imaging from the each cycle using PBMC suspension cell. As you see from the each imaging, there's only minor vibration in each cell's position. There's no cell detachment during the Codex learning. It means that the staining cycles are very stable and gentle to maintain the tissue well. After staining and imaging cycle, Codex produce tons of the images from tile, G-stack, and channel, and cycle. You don't need to worry about this. Codex has very sophisticated software package to manage this huge image data. Codex software efficiently processes the image like drift compensation, deconvolution, stitching, background subtraction, and cell segmentation. Codex software leads single cell analysis data from whole slide with the FCS or CSV format. Finally, you can have the whole slide scan image, which you can see the expression of the each 40 proteins. This is an example of the breast cancer tissue stained with the 26 antibodies. You can see all marker expression in the different color. I will show you one example of the whole tissue staining image from Codex system. You can see the whole tissue imaging images in Codex visualization software. This is a human tonsil tissue stained with the 41 antibodies. Actually, tonsil tissue is a good positive sample to test major immune cell types. This whole slide image shows only the 6 colors for 6 markers because human eyes cannot recognize 41 colors at one time. So the Codex software gives an option to pick the marker for each color. I will explain this option in next slide. After staining the tissue, you can zoom in 1 RY as shown in the box on the right. It's completely high resolution to see the marker expression in single cell and cellular compartment level. The Codex Viewer software has the panel that allow you to combine and adjust the color by marker. So you can select the color of each marker that you want to see, and they can be visualized on the imaging. As another option, it can be switched to IHC image for each marker. It's very convenient to communicate with your pathologist. The biggest advantage of Codex is the information at a single cell level, not only the imaging. Codex automatically segments cells on the imaging and then release localization information and intensity information of the over 40 markers from each single cell at runtime. This single cell based analysis is done automatically during the processing And you can find the exported Excel file, which include all this information in processor folder. Because Codex analyzes each marker's intensity in a single cell level, the Codex software can visualize the gating plot like a cell cytometry. If we want to select some specific cell which co-express two markers, software can make the subpopulation of this cell in the plot. But most important difference of the Codex software from cell cytometry, the Codex software can merge all these selected population on the imaging. And then it can be confirmed by the toggling within image to data. So this is a very useful tool to check the cell selection, cell segmentation quality. If we handle the small number of the markers, the previous gating plot will be enough for analysis. But if you imagine over the 40 markers in single cell tissue sections, thousands of the different cell phenotypes will be available. So to help you unsupervised and various cell phenotyping, Codex software has a great automatic clustering tools. You can just click the X-shift clustering in the menu, then select markers for cell clustering. Then there's some optimizing option for best clustering. You can tune some parameter like a population size and intensity information option. Then you can see the software runs for clustering on the right corner. It's a real-time recorded file during my trial. As you see, it doesn't take long time for analysis. Then you can have many clusters with a specific marker expression with a heat map and the size of the population. If you click the plus at the bottom, all this population will be merged on the imaging. You can see various population with different color. One of the benefits of the Codex is that you can make group with a similar character with a similar color automatically. If you see the imaging, the major luminous cells were grouped with a blue, and then basal cells were grouped with a magenta, and the vascular cells were grouped with a yellow, and T cell with the orange. The reason why we try to identify many different cell phenotypes is our understanding what kinds of the cell functions in tissue and how they communicate for proper response to the immune therapy. It's very important to approach intensive immunology to understand the role of the immune system. So the T-SNP map or U-maps are most common graphic results which we can visualize these various cell phenotypes and their relationship. Codex can make this T-SNP map very easily. You can click the gating in the menu, then find the T-SNP, X-T-SNP in scrolling in X-axis and Y-T-SNP in Y-axis. Codex automatically calculates this T-SNP during the data process. So Codex has the option to change the color based on different population or marker expression. For example, you can find T-SNP population with CD3 or vascular cell with a CD44 and proliferating tumor cell with a Ki67. One biggest difference of this T-SNP of the Codex against the transcriptomic data is that Codex, the T-SNP plot include the spatial information too. It means the closed population in t-SNE map, it means that they are geometrically closer on tissue. Now, I will show you a very useful tool in cell phenotyping visualization in Codex. Codex enables advanced phenotyping, and it can be visualized as a Voronoi diagram in imaging. From the automatic clustering, you will have information about the various cell phenotype, and now you can make your own annotation. In this imaging, almost over 20 phenotypes can be determined from just a single ROI within the tissue. Voronoi map is a very good tool to visualize these various cell phenotype with a very various color. Spatial mapping is another important tool from the Codex multiplexed data. From the previous slide, we found over 20 phenotypes of cells. Codex software showed the convenient spatial map, which means the distance map between different cell phenotypes. From this map, the red means two phenotypes are close, and the blue means they are far from each other. You can confirm this on Voronoi map. In the spatial map, FDC cell is fair green. The FDC cell is close to the lead B cell. In the Voronoi map, the fair green, the FDC cell is close to lead the B cell. If I showed another example, vascular endocellular cell is far from T cell interacting with the DC, and in the spatial map, So let's confirm this in the Voronoi map. You can find the T cell in yellow is far from the magenta, the T cell, the interacting with the dendritic cell. So if you may handle the huge number of the population, so it's not easy to compare the distance between each cell manually. So that's why Codex also supply very convenient and automatic spatial mapping tool to handle huge number of the cell population. So if you click the spatial analysis in the menu, it asks you the minimum and maximum distance option between each cells. Then generate the distance heat map. So red means two populations are close, and the blue means are far from each other. Then you can also see the quantified distance value from each well. So finally, if you click the circles at the bottom, you can find the spatial, useful spatial map. The circles is a great tool to visualize this distance map within the multiple spatial calculation. It shows all cell population in imaging and each markers which define each population and how many cells consist of this population. Most important analysis in the circles map is that it shows the distance between different populations and how many cells are closely interact with each other. I explained about the Codex chemistry, Codex workflow, and analysis tool. Now I'd like to introduce how you can utilize Codex for your research. Most of you may experience in a single cell sequencing to identify the important gene or protein in tumor microenvironment. The site-seq sequencing technology from the 10x Genomics is one of the useful technology to identify RNA and protein from same single cell. This slide shows the concept of the site-seq sequence. Despite of the advantage for single cell-based analysis, this site-seq has a limit in obtaining spatial information. This technology requires tissue dissociation for single-cell isolation. It means it can miss the cellular localization information. Codex is the best tool to compensate the lack of the spatial information from cytosolic sequencing data. Dr. Cameron Group in University of Pennsylvania developed computational algorithm that combine the cytosolic sequence plus Codex data. We call this Stevia. If you use same antibody panel in cytosolic and Codex staining, this single cell for cytosolic will acquire spatial information from Codex through the Stevia. It means that all RNA transcriptome from single cell sequencing also will have a spatial information, and then it can be visualized on imaging. So it's great the advantage to NGS users to have spatial information of each gene in tissue and help your discovery of the specific marker with the localization information. So if you are 10x genomics users, please consider to use the Codex to expand your research. Now, I guess you think why and how spatial cell phenotyping is important in our cancer immunotherapy research or clinical outcome. I'd like to introduce one paper published in the Cell by Dr. Nolan. He is the inventor of the Codex. This is the best paper to show what is Codex and what kinds of data is available using the Codex. This paper shows the multiplexed tissue imaging and the computational analysis framework applied to the colorectal cancer. It allows the integration of the how spatial organization of the immune tumor microenvironment is linked to clinical outcomes. I'm so excited to introduce great opportunity to learn more about this research. The first author of this paper, the Dr. Christian Schulz, will give a talk in this research in FIMSA on 3rd of November. I strongly recommend you attend this session to learn more about how Codex can contribute to predict cancer immunotherapy response from the discovery of the single-cell tumor architecture using the Codex. I also want to introduce Codex Demo Service Program by Akoya Bioscience headquarters. You can see most common immune cell markers on your tissue and test more functional marker with additional panel and your own the specific markers. These Codex panels are tested in several tissue types and have made great result. This is the last slide of my presentation. Thank you so much for your attention. So if you have any questions about Codex or Codex Demo Service Program, please contact BMS, the Korea sole distributor of the Akoya Bioscience. We are always open to collaborate with you. Thank you so much.